As you know from the lectures, phytoremediation is the use of plants to clean up toxic chemicals in the environment. In the next two homework assignments, number 5 and 6, you're going to design a plant that can be used to decontaminate soil that's been polluted with mercury. Now, this is an interesting story, and it all starts with the discovery of gold in the Sierra Mountains in 1848. News of the gold discovery brought thousands of gold seekers to California, known as the 49ers, which of course is the same name as the San Francisco football team. And these 49ers scoured the Sierras, panning for gold in rivers and collecting nuggets right out of the ground. And by the mid-1850s, about 250,000 gold seekers had arrived, and because the easy nuggets had already been taken, the miners started turning to hydraulic mining. They collected water from mountain lakes and redirected it into ever-narrowing channels, eventually through a large canvas hose and out through a giant iron nozzle. This resulted in a very high-pressure jet of water that was directed to the side of mountains, and the gold-bearing slurry that came off these mountains was passed over wooden sluices, and the gold settled to the bottom. Now, this is a really effective way to mine gold, and by 1880s, approximately 11 million ounces, which is today worth about $14 billion, were collected by this hydraulic mining. But environmentally, this is a disaster. Whole mountainsides are blasted away. Enormous quantities of silt and gravel poured into streams and rivers, completely destroying the natural waterways as well as the California agriculture was just emerging at the time. The environmental devastation was further amplified by the way gold was collected in the sluices. Miners added millions of pounds of elemental mercury, it's the same stuff that's in thermometers, to the sluices to help them recover the gold. You see, gold and mercury form a heavy amalgam that sinks to the bottom of sluices while gravel and sand pass over the top. But the process is obviously very messy, and much of the mercury was flushed into the Sierra Mountain streams. And the problem is mercury causes nerve damage. More significantly, microbes in the water convert elemental mercury, which is in the thermometers, to methylmercury, which is an organic form that's readily taken up by biological organisms. But methylmercury is not very easily eliminated, so it bioaccumulates, particularly in fish. Methylmercury is much more toxic than elemental mercury, particularly to developing embryos. An illustrative case of methylmercury poisoning occurred in Minamata, Japan around 1950. For many years, the Chizo chemical factory dumped industrial wastewater into the Minamata Bay. And the wastewater contained methylmercury, and this bioaccumulated in the fish and also in people that ate them. Ultimately, over 10,000 people were crippled by methylmercury, and about 2,000 died. The U.S. Geological Service estimates that thousands of pounds of mercury may still remain at historic mining sites throughout California, as well as the rivers that run through them. The USGS surveys fish caught in mountain lakes, and several have found to have toxic levels of mercury, particularly dangerous for children or women who are considering having children. So in the current exercise, you're going to play the role of a biotechnology consultant who's been hired by the Empire Gold Company to develop plants that can help alleviate the methylmercury contamination problem. Coming from UC Davis, you know the plants absorb minerals through their roots, and similar mechanisms could be used to extract elemental toxins from the soil and transport it to above ground parts of the plant. The toxic-laden plant parts could then be harvested and transported to an ex situ detoxification facility. You're going to use a strategy proposed by Rich Meager and colleagues in a 2005 paper I gave you in assignment number 5. The strategy makes use of two bacterial genes, MER-A, mercuric reductase, and this catalyzes the conversion of ionic mercury to elemental mercury, and MER-B, or organomercury lyase, and this catalyzes the conversion of methylmercury to the less toxic ionic mercury. In the first part of this assignment, you're going to read the USGS report on mercury contamination of historic gold mines, so you know a little bit more about the problem. You're then going to find and characterize the MER-A and MER-B genes. So search the gene database for mercuric reductase. You want to identify the mercuric reductase gene that was originally identified in the archaea Thermoplasma acidophyllum. One way to do that is to look at the results by taxon, the taxonomic group. The organism we're interested in is an archaea, and the thermoplasm we're interested in is in this family, the Thermoplasmataceae. Here it is, Thermoplasma acidophyllum DSM-1728. That's the organism you want. Click on the gene ID. You've seen this before, and there's many questions you'll be able to answer here as far as what the gene looks like using the FASTA file or what the protein looks like using the protein file. Some of the questions concern the Thermoplasma acidophyllum itself. One place to look for that is under the genome link. And what you'll see here is a description of the organism that was isolated from which the DNA was sequenced. Answer a couple questions using this information and using the information from the gene browser. You're also being asked about patents on MER genes. To find these, go to the protein database and use BLAST-P to search the patent database. You'll need to click around a bit to answer the questions on the worksheet, but here's a hint. Patents are good for 20 years after their filing date. The second part of this project, assignment number 6, is to design a transformation vector for introducing these genes into plants. But there's approximately 300,000 plant species, so you need to decide which one to transform. I mean, you want a fast-growing plant, you want it to be able to grow vigorously in contaminated soils, and it needs to grow in the Sierra Mountains. After some discussions with your UCD colleagues, you decide that switchgrass, which is often discussed as a possible biofuel crop, may be a good plant to transform. So to find out more about switchgrass, you need to go to the NCBI database and search all the databases. Some of the questions can be answered using the genome database. You're also going to use PubMed Central to find articles about switchgrass biotechnology. You also need to find 2 additional articles described in the worksheet that relate to switchgrass transformation. You might need to use Biosis for these because they're not medically focused. Figure 1 in one of these papers is reproduced on your homework assignment. Use the figure legend and information in the text to answer the questions about the different gene components. Using figure 1 as a guide, draw your own vector that includes the MER-A and MER-B genes. Be sure to add all the components necessary for a plant transformation vector. You need tDNA border sequences. You need a marker gene for the selection of transgenic cells. You need 2 mercury detox genes and promoters and terminators for all of these, which means 3 promoters and 3 terminators. You also need a gene outside of the tDNA to select for the plasmid in bacteria. You can draw this vector by hand, that's fine, but please make it legible. You should then scan or photograph your plasmid and include it with the downloaded assignment onto SmartSite. That's it for those 2 assignments.